We are talking about the human cell. And I know the moment I say the cell, a lot of people probably flash back to ninth grade biology.
Oh, yeah.
You're picturing a plastic model on a teacher's desk, maybe drawing a wobbly circle and labeling the nucleus. But the source material we have today, this isn't that. This is chapter 2 of a heavy-duty physiology text. This is like the engineering manual for life.
That is exactly the right way to frame it. I'm glad you said that. We aren't just looking at a diagram, we are looking at the machinery. So the goal of this deep dive is to understand the cell not as a static blob of jelly, but as a functioning dynamic metropolis. Because that is what it is.
A metropolis.
It has infrastructure, it has power plants, it has a waste management division, it has a government, and it has strictly enforced borders.
Okay, before we get into the city planning, though, we have to establish the scale, because this number you highlighted in the notes just stopped me in my tracks. How many of these cities are we actually made of?
The current estimate, and it is an estimate, is around 100 trillion cells in the human body.
100 trillion.
It's a number so large it effectively loses its meaning.
It really does.
To give you some context, that is more than the number of stars in the Milky Way galaxy. So you right now are a walking galaxy of cells.
That is humbling and a bit overwhelming. And the text, it mentions a specific breakdown for red blood cells, right?
Yes, and this is a great little trivia point. Of those 100 trillion, about 25 trillion are red blood cells.
So a full quarter of our entire cellular population.
A full quarter is dedicated to strictly one job, carrying oxygen. They are the transport fleet, the FedEx trucks of the body.
Okay.
But that leaves 75 trillion other cells, nerve cells, muscle cells, kidney cells, that form the actual structure and function of you.
And despite how wildly different a neuron looks from, say, a liver cell, the core mission of this deep dive is that they all run on the same basic operating system.
Precisely. They might have different jobs. You know, like a baker has a different job than a banker, but they both need to eat, they both need to breathe, they both need to take out the trash, and they both live in houses with walls. The mechanisms we are going to break down today apply to almost every single one of those 100 trillion cells.
So let's build this city. If we were to take a cell and throw it into a blender, and apologies to any biologists listening, what's the raw material? What is the stuff of life?
The old school term for it, the one you see in the textbooks, is protoplasm.
Which sounds like something from a 1950s B movie.
It really does. The attack of the protoplasm. But in physiology, protoplasm isn't a single goo. It's a collective term for the 5 basic substances that make up the cell: water, electrolytes, proteins, lipids, and carbohydrates.
Okay, let's walk through these ingredients, because I think understanding the soup explains a lot about how the cell actually works. Ingredient number 1, water.
The universal solvent. In most cells, water makes up somewhere between 70 to 85% of the total mass.
That is a huge percentage. So we're basically sophisticated water balloons.
Essentially, yes. Unless you're a fat cell.
Ah, right. There's always an exception.
Adipocytes, or fat cells. In their case, the cell is basically a storage locker for fat, so water gets squeezed out to make room. But for any functional cell, a cell that's actually doing work, water is the medium.
And that's critical.
It's absolutely critical. We tend to think of water just sitting there like water in a glass. But in the cell, it's the atmosphere. It holds the dissolved chemicals. It suspends the particles. Chemical reactions in the body don't happen in a vacuum. They happen in water.
Okay, so we have the water. But if we were just pure water, we'd be puddles. We need a spark. Is that where the electrolytes come in?
That's exactly it. Or ions. This is where the chemistry starts to get really, really interesting. The text lists things like potassium, magnesium, phosphate, sulfate, bicarbonate.
Now, I notice a very specific distinction in the notes here. The text separates intracellular ions from extracellular ions. Why does that matter? Why isn't it just a big mixed-up soup?
This is one of the most fundamental concepts in all of physiology. So the cell works incredibly hard to maintain a difference between the inside and the outside. Okay. Inside the cell, you have high levels of potassium, magnesium, and phosphate. Outside the cell, so in the blood and the fluid between the cells, you have high sodium and chloride.
So potassium inside, sodium outside, the big takeaway.
That's the one to remember. And you might ask, who cares? But that separation is what creates life. Think of it like a dam.
A dam, okay.
If the water level is the same on both sides of a dam, you generate no power. The water just sits there. But if you have high water on one side and low on the other, you have potential energy.
You have a gradient.
Exactly. By keeping potassium in and sodium out, the cell creates an electrochemical gradient. It is literally a battery.
A biological battery.
That battery is what allows a nerve to fire a signal. It's what allows a muscle to contract if those concentrations ever equalize.
The battery dies.
The battery dies, and so do you. That's why your body spends so much energy just pumping ions back and forth across the membrane. It's constantly keeping the battery charged.
That puts a whole new perspective on drinking an electrolyte drink. You're literally recharging the battery fluid. Okay, next ingredient on our list, proteins. The source says this is the second most abundant substance, about 10 to 20%.
Correct. But here, we need to make a really clear distinction, because not all proteins are the same. We have structural proteins and functional proteins.
The name structural seems pretty self-explanatory. These are the building materials, the bricks and mortar.
Think of them as the steel girders, the rebar, and the asphalt of our cell city. These are long, thin filaments. Inside the cell, they form microtubules and the cytoskeleton, which is what holds the cell's shape so it doesn't just collapse into a blob.
Okay, so they provide the form.
Right. And outside the cell, they form things like collagen in your skin or elastin in your arteries. They're static. Their job is to be strong and provide structure.
And the functional proteins, these sound more active.
These are the workers. They are not static. They are mobile. In the text, they're described as tubular globular.
Tubular globular, it's a catchy name.
It basically means they're balled up into these complex three-dimensional shapes, and these shapes are everything. These are the enzymes.
The catalysts, the things that make stuff happen.
Right. If the structural proteins are the factory walls, the functional proteins are the machines on the assembly line. They catalyze chemical reactions, for example, splitting glucose to get energy. Every single thing the cell does is mediated by a functional protein.
Okay, so we have water, the medium, ions, the spark, and proteins, which are both the structure and the workers. Next up, lipids, or as most of us would call them, fats.
Lipids are defined by a very simple chemical property. They dissolve in fat solvents, but they do not dissolve in water.
Oil and water don't mix, the classic example.
Exactly. The main ones we care about in the cell are phospholipids and cholesterol. Together, they only make up about 2% of the total cell mass.
That seems really low compared to water or protein.
It is low by weight, but their function is absolutely critical. Because they hate water, they are perfect for forming barriers. If you're a cell made of water and you're living in a world of water, you need a raincoat to separate inside from outside. That's what the lipids do. They form the membranes.
Unless, once again, you are a fat cell.
Right. The exception strikes again. In adipocytes, triglycerides, which are neutral fats, can be up to 95% of the mass. That's just pure stored energy. It's the pantry stocked up for a long winter.
And finally, carbohydrates. I usually think of carbs as fuel. Is that accurate here on the cellular level?
For human cells, yes. That's their primary role. They have very little structural role, unlike in plants where cellulose, which is a carb, builds the cell walls. In us, carbs are about 1% of the cell mass.
That's it, just 1%.
It's small because it's being used constantly. It's like the coal for the furnace. It exists in the fluid as dissolved glucose, which is ready energy, and inside the cell, we store a little bit of it as glycogen.
Glycogen is the insoluble polymer, the text says.
Yes. Think of glucose as cash in your pocket, ready to spend. Glycogen is money in the savings account. It's a bunch of glucose molecules chained together. When the cell needs energy fast, it just breaks a few links off the glycogen chain, turns it back into glucose, and burns it.
Okay, so we have our ingredients list. We've mixed the protoplasm. Now we need to build a city. And the first thing any city needs, especially in a biological sense, is a border, the cell membrane.
Or the plasma membrane. And this is one of the most elegant, beautiful structures in all of nature.
The source material really emphasizes how incredibly thin it is, 7.5 to 10 nanometers.
It's almost impossibly thin. You would need to stack thousands of them to equal the thickness of a single piece of paper. And yet, this tiny, tiny film manages to control everything that enters or leaves the city.
Let's really get into the physics of this, because I think the how is what's so amazing. The text calls it a lipid bilayer. Let's break that down.
Right. And to understand this, you have to visualize the phospholipid molecule itself. It looks a bit like a balloon with two strings hanging off of it.
Okay, I'm picturing it. A balloon with two strings.
The balloon part, that's the phosphate head, is hydrophilic. Hydro for water, philic for loving. It loves water. It wants to be wet.
Okay.
The strings, those are the fatty acid tails, are hydrophobic. Phobic for fearing. They are terrified of water. They do everything they can to get away from it.
So you have this molecule with a split personality. One end loves water, the other end absolutely hates it.
Exactly. Now, imagine you dump a few billion of these molecules into a bucket of water. what happens? It's not a biological decision. It's pure physics.
The tails freak out.
The tails freak out. They frantically try to hide from the water, so what's the most efficient way to hide? They clump together, pointing inward, and the heads, which love water, arrange themselves facing outward to shield the tails from the watery environment.
They form a sandwich.
A perfect sandwich. The bread is the phosphate heads facing the water on the outside of the cell and the water on the inside of the cell. And the filling is all those fatty tails hidden in the middle, completely dry and happy. This is the bilayer.
And because that middle layer is pure fat.
Water-soluble things cannot get through. Ions, glucose, urea, they get to that fatty core and they just bounce right off. It's an impenetrable barrier for them.
But not for everything. Some things can get through.
No, not for everything. Fat-soluble substances like oxygen, carbon dioxide, and alcohol, they see that fatty middle layer and they say, great, I'm fat too, and they just slide right on through. That is why oxygen gets to your cells so fast, and also why alcohol hits your brain so quickly. It doesn't need a key. It just walks right through the walls.
That's a slightly scary thought about alcohol. But wait. If the wall stops glucose and ions, which you said the cell absolutely needs to survive, how do they get in?
That is the million-dollar question. And that's where the proteins come in. Remember, the membrane isn't just fat. It's studded with proteins. About 55% of the membrane's mass is actually protein.
These are the membrane proteins.
Yes, and we can divide them into two main types. We have integral proteins that punch all the way through the membrane from the outside to the inside.
So they're like tunnels or gates.
Exactly. Some are simple pores, just channels that are selective for, say, water or specific ions to flow through. Others are more complex. They're called carrier proteins.
What do they do?
These are more active. They'll actually grab a substance, like a glucose molecule on one side, change their shape, and physically drag it through the membrane to the other side.
And sometimes they drag it against the current, right?
Yeah.
Uphill.
That's called active transport. It's like pushing a ball up a hill. It requires energy from ATP, but the carrier protein gets the job done. It forces the molecule to go where it doesn't want to go.
There's another type of integral protein mentioned here that doesn't transport anything at all, the receptor.
Receptors are crucial for communication. Imagine a doorbell on your house. You press the button on the outside, and a bell rings on the inside. Your finger doesn't pass through the door, but the information does.
So a hormone like insulin is the finger pressing the button.
That's it. Insulin floats by in the bloodstream. It binds to its specific receptor on the outside of the cell, and that receptor then triggers a chemical change inside the cell that says, hey, we've got insulin out here. Open the gates for sugar.
It's an incredible communication system. And there's one last feature of the membrane I want to touch on, the glycocalyx, which sounds like a fantastical creature from a Harry Potter novel.
It does, doesn't it? But glyco just means sugar. The glycocalyx is a loose carbohydrate coat on the outside of the cell.
So the cell is literally sugar-coated.
Literally.
Mm-hmm.
Most of the proteins and some of the lipids on the surface have these little chains of sugar dangling off them, creating this fuzzy coat.
And what does this fuzzy coat do?
It has several important jobs. First, it usually has a negative electrical charge, which helps to repel other negative objects.
Kind of like a weak force field.
A very weak one, yes. Second, the glycocalyx of one cell can attach to the glycocalyx of another. This is how cells stick together to form tissues. It's like biological Velcro.
Okay.
And third, it acts as an ID card. The specific pattern of sugars is unique, and it tells the immune system, hey, I belong here, I'm part of the team, don't attack me.
So it's a force field, Velcro, and an ID card.
Yeah.
Not bad for a sugar coating. Okay, we have officially crossed the border. We are inside the city. Welcome to the cytoplasm.
The interior space. The fluid part is the cytosol, that jelly-like soup we mentioned earlier, full of dissolved proteins and electrolytes. But floating in that soup are the organelles. These are the functional districts of our city.
And if we're continuing our city analogy, we need to talk about the industrial district, the manufacturing plant, the endoplasmic reticulum, or the ER.
The ER is massive. Visually, imagine a vast, interconnected network of flat, tubular sacs. If you could take the membrane of the ER and unfold it, its surface area would be 30 to 40 times larger than the cell membrane itself.
That is an insane amount of surface area packed into such a tiny space.
Right.
And the notes say there are two neighborhoods here, rough and smooth.
Right. The The rough ER is called rough because under an electron microscope it looks bumpy. It's covered in these tiny granules called ribosomes.
And ribosomes are the protein assemblers. They read the genetic code.
Correct. They take the instructions from the nucleus and stitch together amino acids to make proteins. So the rough ER is the factory floor where new protein molecules are born.
Okay, rough is for protein. So what about the smooth ER?
No bumps, no ribosomes, so it's smooth. Its main job is synthesizing lipids, fats like phospholipids and cholesterol, so easy to remember. Rough for proteins, smooth for fats.
Okay, so the ER is churning out product. But a factory can't just pile boxes on the floor. You need a shipping department. Enter the Golgi apparatus.
The Golgi is fascinating because it works in direct partnership with the ER. Small transport vesicles, little bubbles of membrane pinch off from the ER carrying the newly made proteins and lipids, and they travel over to the Golgi.
And the Golgi acts like the city's main FedEx hub.
It does, but it's also a finishing school. The Golgi processes these substances. It might add some of those carbohydrate chains to them to make them functional, and then it wraps them up in highly concentrated packets.
And these packets have specific names. The text mentions secretory vesicles and lysosomes.
Exactly. Secretory vesicles are packages destined to be shipped out of the city, things like hormones or digestive enzymes that the body needs elsewhere. Lysosomes are different. They stay inside.
Let's talk about lysosomes, because the text describes them as the digestive system of the cell. But when I read about how they work, they sound kind of terrifying.
They are little bags of destruction. They're filled with powerful digestive enzymes called hydrolases.
Hydrolase, meaning they use water to split things apart.
Yes. They can dissolve proteins, glycogen, lipids, basically any organic matter that gets inside them. They are the stomach of the cell. But here's the critical part. They are surrounded by a very tough lipid membrane.
Why is that membrane so important? What's the point?
Because if that membrane breaks, the enzymes leak out and they eat the cell itself from the inside out. It would be like your stomach dissolving your own body. The membrane keeps all that destructive power contained.
So it's a suicide bag, essentially. If the cell is damaged or needs to die, popping the lysosomes is a quick way to do it.
It's a process called autolysis. And yes, it is the cell's self-destruct mechanism.
Now, there's a similar-sounding organelle mentioned, the peroxisome. How is that different from a lysosome?
Two main differences. First, they aren't made by the Golgi. They seem to self-replicate or bud off from the smooth ER. Second, and more importantly, instead of hydrolases, they contain oxidases.
So they use oxygen to do their work.
They do. They combine oxygen with hydrogen ions from various chemicals to form hydrogen peroxide, H2O2.
Wait, isn't hydrogen peroxide poisonous? I mean, I use that stuff to clean cuts, specifically because it kills bacteria.
It is highly reactive and potentially very dangerous to the cell, but the peroxisome uses that danger. It pairs the H2O2 with another enzyme called catalase to oxidize substances that would otherwise poison the cell. It's the city's hazardous waste treatment center.
Can you give me a real-world example of this in action?
The liver. About half of the alcohol a person drinks is detoxified by peroxisomes in your liver cells. They use that hydrogen peroxide to break the alcohol down into something harmless.
So next time I'm at a bar, I should raise a glass to my peroxisomes. They're doing the heavy
Yes,
lifting.
they certainly are. They are working overtime.
Okay, so we have factories, shipping centers, and hazmat teams. But none of this runs without electricity. We need to talk about the power plant, the mitochondria.
Ah, the celebrity organelle. Everyone knows the meme. The mitochondria is the powerhouse of the cell.
It's the one thing we all seem to remember from biology class. But looking at the source material, I feel like the meme really undersells it. We treat it like a battery you just slot into a toy, but the physiology suggests it's more like a separate entity entirely that just happens to live inside the cell.
You are picking up on a very big evolutionary clue there.
Yes. Let's look at the structure first. It has two membranes, right? A double wall.
Yes. An outer membrane and an inner membrane. And the inner membrane is folded into these massive shelves or tubules called cristae.
Why all the folding? What's the purpose of that?
Surface area, surface area, surface area. If you could take that inner membrane and flatten it out, you would have a huge amount of space to attach the oxidative enzymes. This is where the magic happens. Nutrients come in, they react with oxygen on these shelves, and energy is released.
And that energy is bottled up in the form of ATP.
Adenosine triphosphate. We'll dive deeper into the chemistry of ATP later, but for now, yes. The mitochondria's sole purpose is to burn fuel with oxygen and churn out vast quantities of ATP. Without it, the lights go out and the city dies in seconds.
But here's the thing that absolutely blew my mind in the text. Mitochondria are self-replicating.
Yes, this is the Russian doll aspect of the cell.
Explain that. Because if I start lifting weights and I need more energy in my muscles, my cell nucleus doesn't just print out blueprints for more mitochondria.
No, the nucleus doesn't build them.
Mm.
The mitochondria themselves sense the increased need for ATP, and they decide to divide. They split in half, just like bacteria do.
That is slightly unsettling.
It gets even deeper. They have their own DNA. A mitochondrion has a completely different and much smaller set of genes than the nucleus of the cell it lives in.
So are you saying that inside each of my cells, there are these little almost alien creatures with their own DNA multiplying whenever they feel like it, and we just provide the house?
Essentially, yes. The leading theory, endosymbiotic theory, is that billions of years ago, mitochondria were independent bacteria. At some point, one got swallowed by a larger primitive cell, but instead of being digested, they struck a deal. I'll give you infinite energy, you give me a safe home and raw materials.
A billion-year-old rental agreement.
Yeah.
That is absolutely mind-bending. We're basically just hosts to these tiny power plants.
We're a symbiotic colony through and through.
Okay, moving on from our alien overlords to the city's infrastructure. The cytoskeleton.
Skeleton of the cell. But don't just think of it as static bones, it's also the city's rail network.
The notes mention microtubules.
Right. These are very stiff, hollow tubular structures made of a protein called tubulin. They act as beams to hold the cell open and give it shape, sure, but they're also the train tracks. Organelles, even the mitochondria we just talked about, can literally walk along the microtubules to get to where they're needed in the cell.
They can walk.
Using motor proteins, they attach to the organelle and the microtubule, and they ratchet along the tracks, step by step, burning ATP. It's a bustling highway system inside every cell.
And finally, after all that, we reach City Hall, the government, the nucleus.
The control center. It contains the genes, the DNA. This is the master blueprint. This determines the characteristics of all the cell's proteins, especially the enzymes. And since enzymes control all the chemistry, the nucleus ultimately controls the cell's entire destiny.
It also controls reproduction, right?
Mitosis, yes, the process of cell division. But I want to point out a little VIP section inside the nucleus called the nucleolus.
The nucleolus, it sounds like a nucleus inside a nucleus.
It sort of is, visually. But unlike the nucleus itself, it has no membrane. It's just a very dense accumulation of RNA and proteins.
And what is it doing in there?
It is the factory builder. Its sole job is to assemble ribosomes. Remember the ribosomes on the rough ER?
Yeah, the protein-making machines.
The nucleolus builds those machines. Those machines then go out to the ER and build all the other proteins for the cell. So it's the metafactory.
Layers upon layers of manufacturing. It's incredible. Now, I want to take a step back for a second. We've described this incredibly complex city. But how does it compare to other life forms? The source has a section on precellular forms.
This is a great way to understand the sheer scale of evolution. If we look at the hierarchy, we start at the very bottom with the virus.
Which is, it's debatable if it's even alive.
It really sits on the edge. A virus is basically just a packet of nucleic acid DNA or RNA wrapped in a protein coat. It is tiny, maybe 15 nanometers. It has no internal machinery at all. It's a pirate. It needs to board a ship, a cell, and hijack its machinery to do anything.
Okay, we step up from there to bacteria.
Like E. coli. These are much bigger, around 1 micron in size. They have functioning machinery. They have ribosomes. They can make their own energy. But they have no distinct nucleus. Their DNA just floats around inside. They're like a one-room cabin. Everything happens in the same room.
And then we get to the human cell.
The eukaryotic or mammalian cell is vastly larger and more complex. The source notes that an average human cell has a volume about 1 billion times that of the smallest virus.
1 billion times.
It just shows you how much complexity has been added over eons of evolution to get from a simple viral packet of code to the functioning metropolis we're discussing today.
So we have built the city. Now let's talk about daily operations. How does the city survive? First, it needs to import goods, the functional system of ingestion.
Endocytosis, that's the fancy umbrella term for bringing stuff inside.
And the text says there are 2 main ways the cell does this, drinking and eating. Let's start with cell drinking, or pinocytosis.
Pinocytosis involves the ingestion of minute particles or droplets of the extracellular fluid, and this happens in almost all cells all the time.
The source had a crazy stat about macrophages here.
It's amazing, isn't it? Macrophages, which are big immune cells, ingest about 3% of their entire cell membrane every single minute through pinocytosis. They are constantly drinking the surrounding fluid to sample it, to test it for invaders.
How does that mechanically happen? It's not like it just opens a little mouth and sucks fluid in.
No, it's a very sophisticated mechanism. Imagine the surface of the cell has these little indentations called coated pits. They're lined on the inside with a protein called clathrin and also with actin and myosin.
Actin and myosin. Wait, those are muscle proteins, the things that make us contract?
Exactly, they're contractile proteins. When a molecule, like a protein, binds to a receptor in that pit, it triggers a reaction. The pit invaginates, it caves inward. Then the actin and myosin contract around the neck of that pit.
Like pulling the drawstring on a bag.
That is the perfect analogy. They pinch it shut, and the vesicle breaks away and floats into the interior of the cell. And this whole process requires energy. It burns ATP, and it also requires calcium ions to work.
So that's drinking. What about cell eating, phagocytosis?
This is for much larger particles. We're talking whole bacteria, dead cells, dust particles. And importantly, only certain cells can do this, mainly tissue macrophages and some types of white blood cells.
So this is the immune system at work, cleaning up messes.
Precisely. It's a similar process to panocytosis, but on a much more aggressive, warrior-like scale. The particle, say a bacterium, attaches to receptors on the macrophage's surface. Usually the particle has to be tagged first by antibodies, a process called opsonization.
So the antibodies from our immune system essentially paint a target on the bacteria's back.
And once the macrophage sees that target, it attacks. The membrane edges don't cave in. They grow outward. They evaginate.
They reach out and hug the bacteria to death.
A deadly hug is a good way to put it. They surround the particle, the actin contracts to help push the particle inside, and then other proteins pinch off the membrane to form a massive phagocytic vesicle.
Okay, so now we have this big vesicle, this bubble, floating inside the cell, and it contains a live bacterium. That seems dangerous. What happens next? Digestion.
This is where the lysosomes we mentioned earlier come into play. They are the SWAT team. One or more lysosomes will move over, attach to the phagocytic vesicle, and then dump their payload of digestive enzymes right into it.
It creates what the text calls a digestive vesicle.
Yes.
Yeah.
And inside that vesicle, the enzymes just melt the bacteria. They break it down into its constituent parts: amino acids, glucose, phosphates, simple things the cell can actually use.
So it not only eats its enemy, it turns its enemy into fuel.
It's ruthless efficiency. The useful stuff diffuses out of the vesicle and into the cytoplasm for the cell to use.
And what about the stuff that can't be digested? The bones and gristle, so to speak.
That's called the residual body. It's basically the cellular trash. The cell moves this vesicle back to the membrane, fuses with it, and excretes the contents via exocytosis. It takes out the garbage.
There's another form of digestion mentioned here called autophagy, eating oneself.
It sounds morbid, but it's absolutely essential housekeeping. Organelles don't last forever. A mitochondrion, for instance, might only last for about 10 days before it starts to get worn out and inefficient.
10 days, that's it.
They work incredibly hard. So when an organelle is old or damaged, the cell needs to get rid of it. It wraps it in a double membrane structure called an autophagosome, then fuses that with a lysosome and digests it. It recycles the materials to build new, healthy organelles.
It's the ultimate recycling program. Nothing is wasted.
And it's critical for survival. If autophagy fails, the cell fills up with junk and eventually dies. This process is hugely important for preventing aging and disease.
Talk about efficiency. Now, let's trace that production line we hinted at earlier, the synthesis and processing system. We know the ER makes proteins and lipids, but let's follow the timeline.
Okay, let's put the stopwatch on. It starts in the rough ER. A ribosome spits out a brand new protein directly into the ER matrix, the space inside.
And at the same time, over in the smooth ER, lipids are being made.
Right. And as the smooth ER makes new lipids, they get incorporated into its own membrane, causing the ER to grow. To keep it from taking over the entire cell, small transport vesicles constantly break off.
The FedEx truck leaves the factory, loaded with goods.
It heads straight to the Golgi. Now, the Golgi does the finishing work. A key job it has is to attach carbohydrate moieties to the proteins.
Which is a very fancy way of saying it sugars them up.
Yes, exactly.
Yeah.
This is how we get things like hyaluronic acid and chondroitin sulfate. These are the main components of mucus and also the ground substance of bone and cartilage.
So the slime in your nose and the gristly bits in your joints are made in the Golgi.
They are products of the Golgi, yes. The Golgi then compacts these secretions into highly concentrated packets ready for export.
And the source gives a timeline for this whole process.
It's incredibly fast. Synthesis in the ER takes about 3 to 5 minutes. Within 20 minutes, those proteins have traveled to the Golgi and are being packaged. And within 1 to 2 hours, they can be secreted from the cell.
That is amazing. From raw amino acids to a finished functional hormone shipped into the bloodstream in less than 2 hours.
And that shipping mechanism is exocytosis, usually stimulated by a signal like a calcium influx. The vesicle moves to the outer membrane, fuses with it, and dumps its contents to the outside world.
We've mentioned energy and ATP quite a few times now, but section 10 of our outline is all about the currency of energy. Let's really dig in. Why is ATP so special?
Adenosine triphosphate. Chemically, it's a molecule of adenine, a sugar called ribose, and 3 phosphate radicals all linked together.
The triad, ATP.
The magic is in the bonds between those last 2 phosphate groups. The source calls them high-energy bonds.
And also labile. What does that mean?
Labile means fragile, unstable. It means they are incredibly easy to break. And when you break one, when you snap that 3rd phosphate off the end, it releases a huge, immediate explosion of usable energy. About 12,000 calories per mole, which is a lot for a single molecular bond.
So ATP becomes ADP, adenosine diphosphate, 2 phosphates.
Right. The battery is now drained. To recharge it, the cell has to use the energy from the food you eat to force that 3rd phosphate back onto the ADP, turning it back into ATP.
It's a constant cycle, discharge, recharge, thousands of times a second.
Yeah.
Let's look at where that recharging primarily happens. The text mentions the chemiosmotic mechanism in the mitochondria.
Don't let the name scare you. Think of it, again, like a hydroelectric dam.
Okay, I like that analogy.
The cell burns glucose and fat from food.
Huh.
In doing so, it strips hydrogen atoms off of them. These hydrogens are then pumped into the space between the inner and outer mitochondrial membranes. They build up there, creating immense pressure.
So the water level is rising behind the dam.
Exactly. Then the cell allows these hydrogen ions to flow back into the inner matrix, but only through one specific channel. A massive protein machine called ATP synthetase. As the hydrogen rushes through, it spins a literal turbine inside this protein.
A molecular turbine.
A molecular turbine. And that spinning motion physically smashes the phosphate back onto the ADP, creating ATP. It is one of the most incredible machines in all of biology. And it's where 95% of your cell's energy comes from.
Only 5% comes from other processes.
Yes. Glycolysis burning sugar outside the mitochondria gives you a tiny bit, maybe 5%. The real power, the industrial scale generation, happens at the mitochondrial dam.
And what do we use all this ATP cash for? What is the city's budget spent on?
3 main categories. First, transport. Those sodium-potassium pumps we talked about at the beginning. In some cells, like the cells in your kidney tubules, up to 80% of all the ATP they make goes just to running those pumps.
80%, just to keep the battery charged.
It is incredibly expensive to stay alive. The second category is synthesis. Building proteins, lipids, carbohydrates. Every time you forge a new chemical bond to build something, you have to pay for it with ATP.
And third.
Mechanical work. The obvious one is muscle contraction.
Yeah.
But also moving chromosomes during cell division and cellular locomotion.
Which leads us perfectly to the final functional system in our outline, locomotion. How do cells move around? Because they don't have legs?
No legs, but they can crawl and they can swim. The 2 main types of movement are amoeboid and ciliary movement.
Amoeboid movement, like an amoeba, I'm guessing?
Exactly like an amoeba. This is how your white blood cells hunt down bacteria in your tissues. It's a crawling motion.
So how does it work mechanically?
Imagine the cell has a front and a back. The front of the cell projects a pseudopodium, which literally means false foot. It reaches out. Then that pseudopodium attaches to the surrounding tissue using receptor binding.
So it throws out a grappling hook.
A perfect way to think about it, yes. And then, using actin and myosin, the rest of the cell pulls itself forward towards that anchor point.
So it's using the same proteins that are in my biceps to crawl through my tissues.
The very same ones. But here, they're not just contracting, they're actively rearranging the entire cytoskeleton. The cell effectively builds new membrane at the front and dissolves the membrane at the back. It is literally flowing forward.
And how does it know where to go? It doesn't have eyes to see the bacteria.
It has a nose. Sort of. It's a process called chemotaxis.
Chemotaxis.
The cell moves toward a higher concentration of a certain chemical, chemotactic substance. If a tissue is inflamed or infected, it releases these specific chemicals. The white blood cell smells that chemical trail and crawls up the gradient right to the source of the problem.
That is just incredible. It hunts by scent. Okay, what about the other type? Ciliary movement.
This is the whip-like movement. You find this on the surfaces of cells in your respiratory airways and in the fallopian tubes.
These are the tiny little hairs we hear about, right?
Cilia, yes. But they aren't just simple hairs. They are incredibly complex machines. A cilium is an outgrowth of the membrane that's supported by a core of 11 microtubules, 9 doublets arranged in a circle around the outside and 2 single ones in the center.
The classic 9+2 arrangement.
Right. And it moves very differently than the amoeba. It uses a power stroke. It stays stiff and rips forward quickly to push fluid, then goes limp and curls back slowly for a return stroke.
Like a swimmer doing a really messy breaststroke.
Exactly. But imagine thousands of them doing it in perfect sync. In your lungs, they create a constant conveyor belt of mucus, moving dust and bacteria up and out of your airway so you don't get sick.
And what powers that whip? What's the motor?
Protein arms made of a motor protein called dynin. These little arms reach out from one microtubule, grab the neighboring one, and pull. This forces the whole structure to bend. And just like everything else we've talked about, it burns ATP to do it.
It is absolutely relentless. The city never sleeps.
Never. If the city sleeps, it dies.
So we have toured the city. We've seen the walls, the power plants, the factories, the recycling centers, the government, and the transportation systems. When you put it all together, what's the big synthesis here? What's the takeaway?
The synthesis is that the cell is a miraculous, self-sustaining unit. It eats, digests, builds, powers itself, cleans itself up, and moves. It is the perfect machine. And every single physiological function we will ever discuss in future deep dives, whether it's your brain processing a thought or your heart pumping blood, it all relies on these fundamental cellular mechanisms working perfectly.
If the sodium pump in a neuron stops, the nerve signal stops. If the mitochondria in a muscle cell fail, the muscle stops contracting.
Exactly. At its core, all disease is just cellular physiology gone wrong.
Before we go, I want to come back to that provocative thought you mentioned much earlier, the one about the mitochondria.
Right, the idea of precellular life.
The fact that mitochondria have their own DNA and replicate independently strongly suggests that eons ago they were separate primitive organisms.
And that implies something truly profound about what we are.
Are we? Are we really just a massive cooperative colony of ancient, smaller organisms that have all agreed to work together?
In a very real way, yes. We are not one being. We are a galaxy of trillions of symbiotic relationships, all humming along in the dark, keeping the lights on for the whole community.
That is something to think about as you go about your day. To just take a moment and imagine the trillions upon trillions of power plants humming inside you right now.
It's a very busy city down there.
Thanks for listening to this deep dive into the cell. We'll see you next time.